With the background understanding of nucleic acids, let's start to look at the synthesis of DNA. We'll begin in this lesson with an overview of the entire process of the flow of genetic information associated with the central dogma. There are three processes involved in the full expression of the genetic information of the cell. Replication is the process of preparing a duplicate copy of the DNA double helix and occurs prior to cell division. In the process of transcription, one of the DNA strands is used as a template to make a complementary sequence of messenger or mRNA. In the final stage of information transfer, the RNA message is translated into the amino acid sequence of a protein. Note that in each case, the order of residues conveys the genetic information. What is known as the central dogma of molecular biology explains the manner in which this transfer of information occurs, as outlined below. Note that information transfer occurs from left to right toward protein synthesis. A very prominent example of an exception to this general flow is that of retroviruses. These are viruses whose genetic material is RNA, like HIV. In their life cycle, the first step in information transfer is a genetic replication process, catalyzed by an enzyme known as reverse transcriptase. Recall that in transcription, a DNA molecule is used as a template to make an RNA molecule. In the case of retroviruses, an RNA molecule is used as a template to make a DNA molecule. Thus, it is the reverse of transcription. As we look into the details of DNA replication, keep in mind that naturally occurring DNA may be either single-stranded or ssDNA or double-stranded dsDNA, depending on the organism. The DNA genome may also be either linear or circular forms. This means that the process of replication might vary some depending on the organism. Because most of the details of this process were first determined in E. coli, and because that is a simple system, we will focus our discussions on the prokaryotic system.